Hello everyone, Dr. Suresh here, and in this video we'll be talking about the beta oxidation of fatty acids. So in our previous videos we have discussed digested food which is containing lipids will be undergoing digestion and absorption and where they will be stored, and after that what is the fate of these stored lipids, right? So the main pathway that will be related for the degradation of stored fat, that is beta oxidation, when we are in need of energy, these stored fat, especially triacylglycerols in adipose tissue, serving as a source of energy. When they undergo degradation, they will separate into glycerol and fatty acids. These three fatty acids acted by the hormone-sensitive lipase and also the capillary lipases, which promotes the free fatty acid degradation and turning them into beta oxidation and provides energy. And the glycerol will be transported to liver and involved in gluconeogenesis. So we'll see the details of beta oxidation. This process is known as beta oxidation. So in chemistry aspect of lipids, we have studied the classification of fatty acids based on the number of carbons, short chain fatty acids, medium chain fatty acids and long chain fatty acids. So short chain means 4 to 6 carbons and medium chain 6 to 12 and the long chain means more than 14 carbons. Same way, we have seen beta carbon, alpha carbon. How we have seen? If suppose CH2 and this CH2 will be like 10 times and 11, 12 and this is COOH, you can say. And this will be the alpha, this is a functional group, okay, like CH2, COOH. So this is a carboxylic group, okay, and this carboxylic group next to the CH2 which is attached to the carboxyl group, that is alpha, and next to this beta. So here removal or the breaking of two carbons at a time. So the breaking of fatty acid is taking place at the beta carbon. Okay, you see here the numbering alpha, beta, gamma, delta, this will be given next from the functional group. Here the functional group is COOH. The CH2 which is attached to the CH2, the CH2 which is attached to COOH is alpha, and then behind that CH2, that is beta. So here in beta oxidation, the removal of carbons at the beta carbon level. So that's why it is known as beta oxidation. Okay, in beta oxidation, every time 2 carbon units will be removed from the beta carbon. Okay, from the beta carbon, 2 carbons will be removed at a time. Oxidation of hydrocarbon chain occurs by sequential cleavage of 2 carbon atoms. So there are lot many steps involved in beta oxidation. So before that, I'll give you some basic aspects. Beta oxidation mainly takes place in mitochondria of the cell. Okay, when beta oxidation takes place, okay, first in general, when we are in need of energy, adipocytes will be having triacylglycerols, and these triacylglycerols will be split into glycerol and free fatty acid. Okay, and these free fatty acids enter into the cell. Okay, and in mitochondria they will be taking place of beta oxidation. Okay, so beta oxidation is nothing but sequential removal of 2 carbons at a time at beta carbon. So that's why it is known as beta oxidation. And what are all the things involved in beta oxidation? So preparative step, there is all of a sudden fatty acid will not undergo beta oxidation. There should be some preparation. Okay, so to make ready that fatty acid to undergo beta oxidation. So what are all the requirements are there? So the main coenzyme A complex is a molecule which contains B complex vitamin, pantothenic acid and a molecule of beta-mercaptoethanolamine, and also carries sulfohydryl group which is forming thioester bond in the acyl-CoA. Okay, this acyl-CoA is very important making or preparing fatty acid to undergo beta oxidation. To emphasize the function of sulfohydryl group, the CoA is sometimes written as CoA-SH. Okay, so this is the structure of coenzyme A. So this is a beta-mercaptoethanolamine and beta-alanine, pantoic acid and ADP, that means adenine diphosphate. So this is a complete structure of CoA, coenzyme A, or you can write it as CoA-SH. So we'll see the beta oxidation one by one. Preparative step 1, activation of fatty acids. Okay, and then second step is role of carnitine. What is the role of carnitine in transportation of fatty acid into mitochondria? And the preparative step 3 is carnitine acyl transferase, how it will be engaging the fatty acid from outer layer of mitochondria to inner layer of mitochondria, right? So that and all we will discuss, and beta oxidation proper. All these steps involved in beta oxidation. First step we will see the activation of fatty acids. Once the fatty acid is coming from the adipose tissue or from circulation, once it reaches to the cell, okay, it will enter into the cell. This is a mitochondria. Okay, so once fatty acid enter inside the cell, so all of a sudden mitochondria will not allow it to enter into mitochondria again. Okay, from the cell, it needs some carrier mechanism to transport it to mitochondria. So before that, it should undergo some modification. So that will come under activation of fatty acids. Fatty acids are activated to their coenzyme A derivatives. So fatty acyl-CoA. Now here free fatty acid will be converted with the help of coenzyme A to fatty acyl-CoA. When you attach a coenzyme A, it will be converted to fatty acyl-CoA, and these steps, preparative steps, will take place in cytoplasm. But beta oxidation proper will take place in mitochondria. Remember the difference. So beta oxidation of fatty acid in mitochondria produce lot of acetyl-CoA, and these acetyl-CoA enter into TCA cycle and produce lot of reducing equivalents, and these reducing equivalents enter into electron transport chain to provide ATP. So that's why mitochondria, both TCA cycle and beta oxidation, both are taking place in mitochondria. That's why mitochondria is known as powerhouse of the cell. So ATP required here for activation of fatty acid to fatty acyl-CoA. So it will be split into AMP and PPI. Here, though you are using one ATP, but the energy that you are utilizing by hydrolyzing two high energy bonds, okay, two phosphate groups will be released. Okay, so it is equal to 2 ATPs. Okay, and the energy from hydrolysis of PPI drives the reaction forward. Those two high energy bonds are utilized in the reaction. The enzyme thiokinase or fatty acyl-CoA synthetase. Remember, the preparatory step of beta oxidation involves the enzyme thiokinase or fatty acyl-CoA synthetase. Three different enzymes are present, one each for short chain, medium chain and long chain. So for long chain fatty acid there is a different thiokinase, and for short chain different thiokinase, or for medium chain different thiokinase. Small chain fatty acids may also be activated by Transferase enzyme using succinyl-CoA. So that is the difference for small chain fatty acids. Transferase will involve in activation of short chain fatty acids. You see here fatty acid with the help of fatty acyl-CoA is converting into, with the help of fatty acyl-CoA synthetase or thiokinase, it will be converted into fatty acyl-CoA. This is a preparatory step of beta oxidation. So now, as I said, fatty acid enter inside the cell and preparative step is taking place and it forms fatty acyl-CoA. Okay, now mitochondria is here. Then how this fatty acyl-CoA will be entering into mitochondria? So there we need a carrier, and that carrier activity will be taken over by carnitine. Then we will discuss about the second step of this preparation of beta oxidation, that is carnitine shuttle. Okay, and what is the role of carnitine here? Fatty acids are activated in cytoplasm, but beta oxidation is in mitochondria. Then how do we already discussed? Mitochondria is having a strong outward structure, it is having a strong mechanism which will not allow the components simply from outside to inside. Okay, they have two layers of membranes, outer mitochondrial membrane and inner mitochondrial membrane. So it is a tough and rigid membrane structure. So the transport fatty acids through mitochondrial membrane is essential. Then which is the substance helping in transportation of this activated fatty acid into mitochondria? ATP is hydrolyzed to AMP and PPI, and energy from hydrolyze in this reaction. The long chain fatty acyl-CoA cannot pass through inner mitochondrial membrane. Okay, so here the transporter carnitine. So remember here long chain fatty acid. For the transport mechanism of carnitine, okay, medium chain and short chain they don't require this carnitine shuttle, but long chain fatty acids require carnitine for their transportation. Right. So the transporter carnitine involved in transfer of fatty acids, especially long chain fatty acids. Carnitine is a beta-hydroxy gamma-trimethyl ammonium butyrate. Okay, and this can be synthesized from one of the amino acid that we will discuss in amino acid metabolism, how carnitine is synthesizing from the amino acids. So how this carnitine is useful here? Transportation of fatty acids. Okay, long chain fatty acid. The enzyme carnitine acyl transferase CAT1, which is present on the outer membrane of mitochondria. Okay. So the hydroxyl group of carnitine to form acyl-carnitine. So carnitine itself involved in the reaction. So it will be converted. So carnitine will react with fatty acyl-CoA and converted into acyl-carnitine. Okay, this reaction occurs in cytosolic side of inner mitochondrial membrane. So the next one is the fourth step, translocase. So here a protein translocase will carry the acyl-carnitine across the membrane to matrix of mitochondria. So here on the matrix side of the membrane, another enzyme, carnitine acyl transferase. See, Cat1 will be acting on the cytosolic side, okay, and Cat2 will be acting on the mitochondrial membrane side, okay, that is the matrix side. So Cat1 present on cytosolic side, Cat2 present in matrix side, right? So Cat1 involved in making acyl-carnitine, okay, whatever the acyl-carnitine is formed in the cytosolic side of mitochondrial membrane will be transported to matrix of the mitochondria by Cat2 by the enzyme carnitine acyl transferase 2. Now after it will be transporting, carnitine will be removed from the acyl-carnitine and regenerated carnitine will again send back to carry one more acyl-CoA. So this acyl-carnitine, as you remove carnitine, this will be again accepts the coenzyme A and form acyl-CoA. So creatine is written cytosolic side by translocase. So in the diagrammatic representation I am showing here, you see here, this is mitochondrial membrane, okay, outside, okay, this is cytosolic side, this is matrix side. So Cat1 is present on the cytosolic side, okay. First, whatever the activated acyl-CoA reacted with the carnitine, so CoA will be removed and acyl group will be, acyl fatty acid will be attached to the carnitine to form acyl-carnitine. So through this carnitine acyl transferase, okay, this acyl-carnitine will be transported inside and there by the enzyme carnitine acyl transferase 2, okay, carnitine will be regenerated back and again sent to cytoplasm by translocase. This is translocase, okay, and anyhow CoA is there inside in the matrix of the mitochondria. This CoA attached with the acyl part and to form acyl-CoA. So acyl-CoA is a starting substance of beta oxidation of proper. So this is the overall diagrammatic representation of carnitine shuttle. So clinical applications of this carnitine shuttle. Medium chain and short chain fatty acids do not require carnitine shuttle. As I mentioned earlier, so they can easily be transported across the mitochondrial membrane but not the long chain fatty acids. So this carnitine shuttle is mainly required for transportation of long chain fatty acids into the inside of the mitochondria. So medium chain easily oxidized. Carnitine deficiency is reported in preterm infants in whom impaired fatty acid oxidation is noticed. So more glucose is utilized resulting in episodes of hypoglycemia. So if in the newborn babies, so if these long chain fatty acids are not crossing over the mitochondrial membrane because of the deficiency of carnitine, there will be shortage of energy. So more glucose will be utilized and more glucose means glucose levels will be coming to below the normal level, leads to a condition hypoglycemia. So carnitine, what is the significance of carnitine? Carnitine is synthesized in the liver and kidneys from lysine. So this is the amino acid which is involved, lysine as well as methionine. Okay, carnitine is synthesized from the amino acids such as lysine and methionine. During growth or pregnancy, the requirement of carnitine might exist in its natural production. Human genetic disorders affecting different steps of carnitine metabolism will cause deficiency of fatty oxidation. That is here Cat1 and Cat2. In some of the genetic disorders, Cat1 may be deficient. So long chain fatty acids cannot be transported into the mitochondrial membrane. Okay, and sometimes Cat2 will be deficient. Though it will be ready on the cytosolic side, inside it will not allow acyl-carnitine. So one or the either way. During the aging process, carnitine concentration in cells diminishes, so people will not be able to get energy from the oxidation of long chain fatty acids. Bones are particularly affected adversely, leading to osteoporosis in elderly subjects. So administration of carnitine is capable of improving the clinical condition. The classical presentation of primary carnitine deficiency is hepatomegaly, elevated transaminases and hyperammonemia. Why? Because hepatomegaly and elevated transaminases, so lysine and methionine are involved in making of carnitine, as carnitine is not making more these accumulation of these amino acids and they will be leading to elevated like ALT, AST transaminases. So it leads to condition because this synthesis is mainly taking place in liver. So transaminase elevation is also indicating carnitine deficiency and hyperammonemia is also one of the reasons. The cause for secondary carnitine deficiency are organic acidureas and drug induced like valproic acid and judavudine. They inhibit the synthesis of carnitine from lysine and methionine. So clinical applications. What are the clinical applications related to the carnitine shuttle? First thing is deficiency of translocase. So what is the role of translocase? So once acyl-carnitine enter into the matrix side of the mitochondria, there after donating its acyl part to the CoA, the regenerated carnitine has to travel back to the cytoplasm for carrying another long chain fatty acid. But here, because of the absence of translocase, the liberated carnitine on the matrix side will not be getting back with the cytoplasm. So that is the thing. It leads to a defective metabolism of long chain fatty acids. So there is no regeneration of carnitine in case of deficiency of translocase. In this condition, muscle cramps are precipitated by fasting and exercise, and high fat diet will be having. So inherited CAT1 deficiency affects only the liver, resulting in reduced fatty acid oxidation and ketogenesis with hypoglycemia. CAT2 deficiency affects primarily skeletal muscles, and when severe, the liver also will be suffered. The sulfonylurea drugs, glibenclamide and tolbutamide, used in treatment of type 2 diabetes mellitus, reduces fatty acid oxidation and therefore hyperglycemia by inhibiting CPT1. So the sulfonylurea drugs were mainly in treating of diabetes. These drugs, what they do, they reduce the beta oxidation of fatty acids, and results in the hyperglycemia by inhibiting CPT1. This is a side effect of sulfonylurea drugs. Acetyl and acyl groups are different, so this difference has to be noted. Acetyl-CoA is a combination of acetate or acetic acid, with coenzyme, and acyl-CoA means acyl group combined with coenzyme A. So acetyl-CoA, that means acetic acid is attached here with the coenzyme. Acyl-CoA means fatty acid attached with the coenzyme A. So know this difference. Now, after completing the preparative steps 1, 2, 3 and 4, now the beta oxidation proper will be taking place here. So 4 reactions are sequentially repeated for complete oxidation of fatty acids. This is called beta oxidation proper. After one round of 4 metabolic steps, one acetyl-CoA unit is split off and acyl-CoA with 2 carbon atoms less is generated. So that's what we discussed. Beta oxidation proper in the sense sequential removal of 2 carbons, and the removed carbons will be liberated as acetyl-CoA. So suppose 14 carbon compound is going beta oxidation. So I will write like this: CH2, CH2. I can write CH2 10 times, so 11. CH2 13, 11, 12, CH2, COOH. So 14 carbons are there. So beta oxidation means this part will be removed here. So 2 carbons you can say. So the 2 carbons will be less from the 14. So what happened here? Again functional group will be attached here. This CH2, CH2, COOH. And this CH2 will be removed as COASH. 2 carbons are here, acetyl-CoA. Right. So this would undergo the same series of reactions again until the fatty acid is completely oxidized. It is a cycle. So until all the now in first cycle, out of 14 carbons, 2 carbons removed. So 12 left. Again one more cycle. So 2 more carbons. Now 10. So like that until the last 2 carbons will be revealed, or liberated, this cycle keeps on going. Right. So finally, at the final step, you will get 2 acetyl-CoA instead of acyl-CoA. First cycle, you will get 1 acetyl-CoA, and 1 acyl-CoA. And this acyl-CoA keep on going repeated cycles until you form 2 acetyl-CoA. So this is a repeated cycle. You see here step 1. So beta oxidation proper has got 4 steps. 1, 2, 3 and 4, and we will study one by one what are the steps are taking place here. So first thing is fatty acyl-CoA undergoing dehydrogenation reaction. So the enzyme is acyl-CoA dehydrogenase. The enzyme is acyl-CoA dehydrogenase. Fatty acyl-CoA dehydrogenated to trans-enoyl-CoA. Trans-enoyl-CoA. You see here, this is the first carbon, second carbon. Here the splitting. CH2C11O SOA. So this is called a fatty acyl-CoA. See, these are the number of carbons. Rest of the carbons. So here FAD is the acceptor of the removed hydrogens to form FADH2, and which is equal to 1.5 ATPs. Note down this calculation. So the enzyme it is removing hydrogens from fatty acyl-CoA. So that's why it is known as acyl-CoA dehydrogenase. What it will form? After donating 2 hydrogens, acyl-CoA will be converted into trans-enoyl-CoA. You see here, 2 hydrogens, whatever is there, it will be donated. Double bond has been formed. The second step is hydration. First step is dehydrogenation. Second step is hydration. Here, the trans-enoyl-CoA accepting a water molecule converted to hydroxyacyl-CoA. The water molecule has been split into OH and H. Okay. So at the double bond point, okay, CH will be converted to CH2 and OH will be added here, right? So fatty acyl-CoA converted to trans-enoyl-CoA by dehydrogenation reaction, and trans-enoyl-CoA again converted back to beta-hydroxyacyl-CoA by hydration reaction. Here the enzyme is enoyl-CoA hydratase. You are hydrating enoyl-CoA. So that's why the enzyme is enoyl-CoA hydratase. So it will form beta-hydroxy fatty acyl-CoA. The L isomer alone is formed during the hydration of the trans double bond. The third step is again dehydrogenation. Okay, here in first reaction what we got? FAD we got. In third reaction, in dehydrogenation reaction, we will be getting NAD. Okay, so the beta-hydroxy fatty acyl-CoA is again oxidized to form beta-keto fatty acyl-CoA. So first is fatty acyl-CoA and to form enoyl-CoA, enoyl-CoA to hydroxyacyl-CoA. Okay, and hydroxy to ketoacyl-CoA. Hydroxy is converting into keto form, right? And this dehydrogenase acts only on L isomer. Here the enzyme is beta-hydroxyacyl-CoA dehydrogenase. Okay, beta-hydroxyacyl-CoA dehydrogenase. NAD accepts the 2 hydrogens and liberated as NADH. So how many ATPs you will be getting? 2.5 ATP. One FADH2 produces 1.5 ATPs and one NADH H+ produces 2.5 ATPs. So finally it is forming beta-ketoacyl-CoA. So 3 steps we have completed. Now fourth step, cleavage. Okay, that means ketoacyl-CoA is supposed to be splitted. Okay, beta-fatty acyl-CoA has to be splitted. So here the enzyme is thiolase. Okay, thiolase is the main enzyme which cleaves the beta-keto fatty acyl-CoA. So it is called Thiolytic cleavage. So splitting of one molecule of acetyl-CoA, leaving behind fatty acid with 2 carbon atoms less. So here you see here, this is functional group alpha and this is beta. So this part will be removed. Okay. So this is CH3, C double bond HCOA, acetyl-CoA, and this will be entering into T-cell cycle and produces 10 ATPs. Right. And rest of the structure acyl-CoA with short of 2 carbons. Okay, and further cycles until it gives 2 acetyl-CoA, 1, 2, 3, 4, depends on the number of carbons. Okay, so suppose I take example of palmitic acid. Palmitic acid is having 16 carbons. Okay, so how many cycles is supposed to undergo? It's supposed to undergo 7 cycles, 7 times it will undergo beta oxidation. So every time it is forming 7. So at the last step it is forming one more extra acetyl-CoA. So 7 plus 1, totally 8 acetyl-CoA it will be producing. 16 carbon palmitic acid. Palmitic acid will be producing total 8. So how many steps are there? 4 steps are there. One is dehydrogenation step. Where FADH2 involved. Okay, next is hydrolysis. There is no energy involvement. Third is again dehydrogenation. Where NADH2 involved. Okay, so 8 acetyl-CoA entering into TCA cycle. So each acetyl-CoA entering into TCA cycle, that means one acetyl-CoA produces 10 ATPs. So 8 into 10. So 80 ATPs are there in case of palmitic acid. Still not yet over. Calculation is still on. Okay. So one FADH2 is producing how many ATPs? It is producing 1.5 ATPs. So how many cycles? 7. So 7 FADH is producing. And NADH2 how many it is producing? 2.5. So how many NADH? 7. So 7 into 1.5 and 7 into 2.5. So 7 into 1.5, 7 into 2.5. So the calculation, how many ATPs it is getting? 7 into 5, 3. So 13 and 7 into 2.5, 14 and 15, right? So like this calculation you have to make out, okay, irrespective of the number of carbons. If suppose if I give you 18 carbons, so like this calculation, so 2 carbons extra from 16 carbons, so it will make, instead of 7 cycles, 8 cycles of beta oxidation, and it will produce, instead of 8 acetyl-CoA, it produces 9 acetyl-CoA. So this is the calculation part, how to done with the beta oxidation. So the newly formed acyl-CoA will sequentially undergo further cycles, steps 1, 2, 3, 4 of beta oxidation until the fatty acid is completely converted to acetyl-CoA. Okay, and like what to say, this is the way how beta oxidation is take place in energetics. Remember, if I'll give you the example of beta oxidation of palmitic acid. So totally according to the old calculation, if NADH is 3 ATP, if FADH is 2 ATP. So according to this calculation, I'll tell you, 16 carbon palmitic acid. So 7 cycles are happening. So totally 8 acetyl-CoA producing. So 8 acetyl-CoA undergoing 12 ATPs based on 8 into 12. So it produces 96 ATPs. Okay, and apart from that, 7 FADH2, 7 into 2, it produces 14. NAD, it will be producing 7 into 3, 21. So it will be producing how many? 6, 4, 11 and 12, 131. So but you remember, during activation of fatty acid in the beginning, preparatory steps, once fatty acid enter inside the cell to make it active, to convert fatty acid into fatty acyl-CoA, there we have invested 1 ATP which converted AMP, but it will be counted as 2. So 131 minus 2, the net production will be 129 ATPs. So it's up to you. I have taken here old calculation. 1 NAD equal to 1 NADH is equal to 3 ATPs, 1 FAD is equal to 2 ATPs. But new calculation is 1 NAD is 2.5 ATPs, 1 FAD is 1.5 ATPs. So it's up to you which calculation you are taking. So that's all about the beta oxidation of fatty acids. Thanks for watching. Thank you.